Okay guys, so we're going to continue on with gene expression today. So we already talked about the chromatin last time, the heterochromatin, the euchromatin. So there are some transcription factors that function as repressors. So what this means is that we can have some transcription factors that are actually going to influence whether the DNA is going to be heterochromatin or euchromatin. In other words, if it's heterochromatin, our DNA is not going to be used to get to our final destination of a protein because it's so tightly coiled. So there's not going to be much gene expression there. But if it's euchromatin, we will have some gene expression. Okay, so we're good here. So let me explain this diagram here. So every single one of our cells, no matter what it is, we have the same DNA in those cells. Whether it's a brain cell, a heart cell, a lung cell, whatever. It's all going to have the same potential to make and express certain genes. So every single one of our cells could make the same proteins to do the same actions as other cells. But obviously, like here we have a liver cell and a lens cell. The liver cell is going to make different proteins than the lens cell. Lens cells are obviously in your eyes. All right, so they're going to each have different proteins because these cells function completely differently. So all of our control elements for these two, they're going to be on the enhancer, which is right here. All right, and if you remember, we have the activators from last video that are going to bind to the enhancer, and then with that we are going to, if you remember, use the mediator proteins and protein bending here, and that's going to allow us to transcribe some of our DNA to get it into mRNA. So both the liver and the lens cell, they have the ability to express these different genes. We have two different genes here. We have albumin and we have crystalline. So if we look here, we have for our liver cell the albumin gene being expressed based on the red, the gray, and the yellow activators. But the crystalline is not expressed. Reason being is we have completely different enhancers and activators upstream from the genes. On the other side, we have our lens cell, which is not expressing the albumin, because again, different activators, different enhancers. So it's going to express a different gene called the crystalline gene. So both of these could potentially make RNA for the albumin gene and the crystalline gene. But the activators that are specific to that cell are the ones that are going to play the biggest role in making sure that, okay, the liver cell is going to make albumin. The liver cell has no reason to make crystalline, so, why make it? Why use that energy to make crystalline when we're not going to use it? That's for the lens cell. So, moral of the story, guys, is every cell can make and express every gene if it wants to. But a lot of cells, there's no reason to express those genes. So the activators are going to be different to make sure that the genes that it needs are the ones that are getting expressed. All right. So prokaryotic operon, guys, like I said, we were going over that one, went over the lac one, went over the tryptophan one. And I told you they were very, very easy. They're very basic. They're in prokaryotic cells. They're in bacteria. The eukaryotic one is a lot more intense. So I didn't want to go over that because we don't really need it. But for our eukaryotic ones, each of our gene has a promoter and control elements. So in the prokaryotic cell, we were just worrying about expressing a specific gene, whether it was the TRP one or the LAC one. And that was pretty much it. For eukaryotics, each one of our genes has a promoter and control elements, and that's going to influence what genes are being expressed in this very big, long chain of DNA. Yeah. So that's pretty much the end of transcription. There are some things that happen after transcription to still regulate our gene expression. and we're going to go over a few of them right now. So they're all going to deal with RNA. So the first one, and we already talked about this last chapter, we just didn't give it a name, is alternative RNA splicing. So what we're going to do here is we are going to have our mRNA produced from our DNA. So we have our DNA that has all the instructions in it. We took it to mRNA, which is pretty much carrying the message of DNA. And this alternative RNA splicing is where we are going to get rid of our introns and keep our exons in the picture. We could go two different ways with this. While we're processing our RNA, we can either go split out our introns and have our exons in there and go to translation, or if there's a problem in it, let's degrade our mRNA. Also, another reason we can degrade it is maybe our mRNA has been used so many times already to make proteins. It's getting old. Maybe there's some mistakes in it. Let's get rid of it. Let's make a nice new fresh copy of RNA. We could do that as well. Here is just an example, and I know we went over this last chapter. The colored ones down here, the red, the green, the purple, the red. So those are going to be our exons. They're kept in. And these other areas here, they're like the peach, light pink color. Those are going to be our introns that are going to be spliced out. I already started talking about this one, but mRNA, we can use it over and over and over again if we want to go from transcription to translation to make various proteins. Now, when we do that, we are going to have that mRNA surviving longer. And in eukaryotic cells, it does survive a little bit longer than prokaryotic cells. But after a while, what we can do is we can degrade the mRNA. All right, so in our mRNA, there is an area at the 3' end, right? It's an untranslated region. So we can have that, and that'll tell us, okay, how long are we going to use this mRNA before it does get degraded? All right, we're going to skip that one. So when the mRNA gets degraded, what's going to happen here is the cell is going to attach to a molecule. It's called ubiquitin, and this triggers the destruction of that strand of genetic material. So now we're going to go over some different non-coding RNAs. So far we had our introns, we had our exons, and we took our exons and we put them to our RNA, to our ribosome, and then it got translated into a protein. Now we're going to go over the different regions of RNA that aren't going to be used to make amino acids, and these are called non-coding RNAs. Our first one, it's going to be a single-stranded RNA. It's called miRNA. It's microRNA. So what these are going to do, these are going to degrade the mRNA, or, and you'll see the picture in the next one, block the mRNA from being translated. The main thing you got to remember here for the miRNA is that it's single-stranded, and that plays an important role because mRNA is also single-stranded. So I know I just threw a lot of letters there, but miRNA is single-stranded as long as the mRNA being single-stranded. So here's our two different paths we could take, and here is the mRNA. Now, in red, we have our mRNA. Okay, so the red one, that's our mRNA there. What we can do is we can bind this miRNA. Sorry guys, I'm trying to enunciate my words so we're not confusing miRNA to mRNA. But if we can bind our microRNA to our messenger RNA, bind it together, what that's going to do is the messenger RNA, is not going to be able to go to the ribosome. If it's not able to go to the ribosome and get translated, we are not going to have any amino acids formed there, just because it has now went from being a single-stranded RNA to a double-stranded RNA. So our next type of our non-coding RNAs is called siRNA. Now, microRNA and siRNA are very, very, very similar. So here is our difference between them. So if we have our DNA, we're going to take our DNA, we're going to make it into our mRNA. So then we will have our nice strand of mRNA, and it was red in the previous slide, so I will just make it red as well. So for our micro mRNA, what we're going to do is we are going to have a few nucleotides, and they're going to be able to bind to our mRNA, and the rest might not, okay? So this area right here will be complementary, meaning if this is a U up here for our mRNA, it's going to be an A. If this is an A, it's going to go to U, G, C, you know, that kind of thing. These ones down here, not necessarily are going to pair up the rest of the way down. That is our microRNA. Over here is our small interfering mRNA, and that one is going to be complementary to our mRNA. All right, so the microRNA, we're just going to have a small region that is complementary, and the siRNA, we're going to have a larger region that is complementary. All right, so it's just a non-coding, non-protein coding strand of RNA, and when it's transcribed, we get this small RNA, this siRNA, and it has some type of function to it. And that function for this one would be to obviously block our translation from occurring. So that's siRNA. All right. So I'm not going to post that video, guys. It goes into a lot of other stuff, and I think just a little picture I drew is good enough instead of going over all that. All right. So just going on, guys. This one talks about the siRNA as well. We are going to overall make a double-stranded DNA. And in siRNA, it can start off double-stranded. MicroRNA is just going to be single-stranded, but siRNA, it can start off double-stranded. And those strands, once they separate, are able to completely complement whatever the sequence of mRNA is, whether it's the coding strand of DNA or the complementary strand. All right, so that's the siRNA. The other way here a little bit. Okay, so our next one is called piRNA or peewee RNA. So what this can do is this can change our heterochromatin to euchromatin or vice versa. In other words, we can reestablish methylation. And if you guys remember, if we add methyl groups, that is going to make our chromatin become heterochromatin. But it can reestablish our methylation patterns during gamete formation. And what this can do is it can block the expression of transposons. Now, we haven't gone over transposons yet this year, but transposons are just DNA sequences that can change its position in the genome. So it might go from one chromosome to another, but you would think that's kind of bad, a transposon. Sometimes it is. That sometimes can create mutations, but at the same time, if it creates them, it can also reverse mutations as well. All right. So all that RNAs do is they change the methylation patterns in the gametes. Okay. Oops. All right. Next section. So our next section is going to be, again, on gene expression, but we're going to talk about specific genes in this one rather than going over the whole picture. So to discover which genes are expressed by cells, we're going to identify which mRNA are being made into the proteins and which ones are not being expressed at all. So a few different ways we can actually do this. So we have gone over the whole process of this. We've talked about going from mRNA to an amino acid. But how do we actually figure out which genes are being expressed on a DNA level? In other words, guys, like, you can't see this stuff. It's so tiny that we're not going to be able to just like, oh, look, there's an A, there's a T, there's a G. You know, we can't see it. So our first way, we have nucleic acid hybridization. So we are going to have one strand of DNA, and we're able to detect what the mRNA would be on that complement. And we're going to do this by using something called a nucleic acid probe. And it's going to, and I know I've showed you guys karyotypes in the past that have fluorescence on them, but we're going to use a fluorescent tag to show us what everything looks like. So there's a process called FISH, F-I-S-H, just like the fish. It's called fluorescence in situ hybridization. And what this does is it allows us to see which mRNA is intact in the organism. And this is what it looks like. Yes, it actually does look like a fish, which is why they kind of use that abbreviation. Another one is called reverse transcriptase-polymerase chain reaction. I know we went over PCR in, I think, chapter 13 it was, where we can get a sequence of DNA and replicate it over and over and over and over again. So reverse transcriptase would be the exact opposite. What we're going to be doing here is we are going to be given a mRNA strand. And if we are going through reverse transcriptase, we're going to take that mRNA strand and bring it back into DNA. So this will give us the DNA sequence of that mRNA. Now, what's nice about this is if we can get that mRNA to a certain point where the exons are still in there and the introns are cut out, it's really nice because we are only going to be seeing the area of our mRNA that's going to be coding for specific proteins and specific genes for that particular cell. So with PCR, well, reverse transcriptase PCR, the DNA that we are synthesizing, it's a complementary DNA, but remember, guys, it's not going to be our full DNA. So just keep that in mind. Here's the whole process here. All right, so we have our mRNA. Once again, we start off with our primer, all right, just like we do in DNA replication, okay? And we are going to be able to form our DNA strand from our mRNA. All right. And then at the end, guys, what we can do is we can run, excuse me, gel electrophoresis to see how long these strands actually are. So last thing in this chapter is going to be how can we use this stuff to our benefit? Well, there's a process called DNA microarray assays. So what this is we are going to use the cDNA that we gained from our reverse transcriptase PCR. And what we're going to do is we are going to make some single-stranded DNA fragments from the mRNA. Now, we're not just going to do this with, every single person. We got to have some kind of goal in mind. So what we can do is we can use the cDNA for patients with cancer. So scientists can look at this cDNA that was made from our mRNA for a bunch of, let's say, cancer patients. I'm just teaching with the lights off. We're almost done here. The scientists then can compare these genes between patients with the cancer, whether they're recovering or not recovering from the cancer. And what they can do is see, okay, we got these cDNAs, because this is pretty much just the genes being expressed. How can we use this for other patients who maybe don't have these genes being expressed and actually put the cDNA in their body, and hopefully that will help them fight the cancer. All right, so some patients are fighting cancer, some of the other patients are not. And in comparing these DNAs, we might be able to figure out why some patients are and some patients are not. So here's a microarray. We got a bunch of different wells here. Now the red well, they are genes that are being expressed in the first tissue. All right, the green one, genes are being expressed in another tissue. The yellow one, these genes are being expressed in both of the tissues that were used as a comparison. Now, if there is nothing in it, nothing at all in the well, this means that the gene is not being expressed in either tissue. So if we were looking for genes whose expression was inhibited by like some kind of hormone treatment, let's say. So again, let me just repeat myself. If we're looking for genes whose expression is inhibited by whatever hormone treatment we're trying to do, we would be looking in areas where there is no fluorescence, no color at all. That's where these genes are not being expressed. Okay. So another way we can do a microarray, so like a different way, we could sequence the complementary DNA samples from different tissues to discover what genes are expressed. So this is going almost like the opposite way. This is RNA sequencing. All right, this one is a little bit better because if we're going from DNA to RNA, in other words, just doing regular transcription, it's a lot cheaper to do than trying to go through reverse transcriptase. Okay, guys, that's all we got to do for this chapter. So that's the end. Go ahead, start studying for your test for gene expression. I know it's a lot of information. Make sure you go over the operons in pretty, big details there with the repressors and everything. There's a lot of variables into them. And like I said, the prokaryotic one is a little bit lighter or easier than the eukaryotic one, which is why we just do operons with the prokaryotes. All right, guys, have a good rest of your day. I'll see you next time.